Hello everyone! Nowadays, vaccination is important to protect ourselves from disease. Disease control requires protection from induction of immunity taken from a population that usually has been killed or weakened. This can be achieved through large-scale immunization programs to achieve herd immunity that capable of inducing a long-term protection, also known as adaptive immunity. Vaccine-induced immune effectors are antibodies produced by B lymphocytes that are able to attack toxins or pathogens in the body. Have you ever wondered how vaccine works? Enough for the introduction, as we will get deeper into how vaccine stimulates the production of protective memory B cells. This begins with the administration of weakened pathogens in vaccine injection. As a result, it causes the primary acquired immune response to start. Antigens that bound to the MHC class II of dendrit cells are transported to the location of activation pathogen-specific naive T cell, which is draining lymph node, DLN. As a result, activation of a B cell occurs due to various types of effector T cell that proliferating in the lymph node. THH cell, which is one of the effector T cells, will bind with the B cell. This correspondence will cause the production on a CD40 ligand of T cell. Then it will bind to the CD40 of a B cell. Cytokines will release from helper T cell to the B cell. This conjugated T cell, B cell, will migrate to the medullary cortex from T cell area. Both cells begin to proliferate and differentiate into lymphoblasts. Some lymphoblasts will remain and develop into plasma cells in the medullary cortex. Meanwhile, some of them will migrate and proliferate into the B cell's primary follicles, forming a germinal center. In here, centrocytes which have highest affinity antigen receptors will be chosen during affinity maturation process. Some of the plasma cells derived from the centroblast differentiation will remain and create antibodies in the lymph node. Meanwhile, other plasma cells will migrate to the bone marrow. As the infection fades, the centrocytes transform into long-lived memory B cells consist of high-affinity antigen receptors which can help in preventing future infection and remain mostly dormant until re-exposed by an upcoming confront with the same antigen.